IB Bio, Transcription Translation Part 2, will focus on the details of transcription and RNA processing. The essential ideas are: genetic information in DNA can be accurately copied and can be translated to make proteins needed by a cell, and information stored as a code in DNA is copied into mRNA. Transcription is the first important step in gene expression, where DNA is copied into a complementary anti-parallel strand of RNA by the enzyme RNA polymerase. This occurs in the nucleus of eukaryotic cells, but would occur in the cytoplasm of prokaryotic cells. The second important step in gene expression is when the code carried by the mRNA is translated into an amino acid sequence by the ribosome. This occurs in the cytoplasm of all cells. In this diagram, we can see the process of transcription and the process of translation, including the ribosome, but we also see an intermediate step called RNA processing. In this step, small sections of the mRNA are removed. Stay tuned. Here's the first IB syllabus statement for this movie. Explain DNA transcription in terms of the formation of an RNA strand complementary to the DNA strand by the enzyme RNA polymerase. RNA polymerase, seen here in pink, breaks the hydrogen bonds holding the two strands of DNA together, causing the molecule to unzip. RNA polymerase proceeds to synthesize a new strand of RNA that's complementary to the template strand of DNA. The section of DNA being transcribed would be called the gene. The RNA polymerase is moving left to right in this image. Once a section of DNA, the gene, has been transcribed, the hydrogen bonds reform between the DNA strands, and the newly formed RNA molecule is released. I'll show you many images of transcription. Choose those that are clear to you for your study. Here we see newly formed RNA being synthesized in a 5-to-3 direction along the antisense strand of DNA by the enzyme RNA polymerase that is both unzipping the DNA and synthesizing the new RNA molecule. The new nucleotides are added to the 3 end of the growing polymer. RNA is synthesized in a 5-to-3 direction. RNA polymerase, seen here, does all of the work in transcription. RNA polymerase unwinds and rewinds the DNA, breaking and reforming hydrogen bonds. RNA polymerase recognizes a region at the upstream end of the gene called a promoter. This is where transcription starts. And RNA polymerase synthesizes RNA in a 5-to-3 direction. Remember that RNA has the base uracil instead of thymine, so if the DNA nucleotide is adenine, then the complementary RNA nucleotide will be uracil. We can see that here and here. In this image, we can nicely see the relationship between the DNA antisense template strand and the newly forming RNA polymer. The RNA is synthesized in a 5-to-3 direction, anti-parallel to the template strand, which is oriented 3-to-5 relative to the RNA. As well, we can see the complementary base pairing between the template strand and the RNA, where uracil is found on the RNA instead of thymine. So here's a relevant IB syllabus statement. Deduce the DNA base sequence for the mRNA strand. The sense strand of the DNA has the same base sequence as the mRNA, with the exception that uracil is in place instead of thymine. The antisense strand of the DNA is the template strand, and the antisense or the template strand is the strand that's transcribed into mRNA. This image nicely shows the arrival of the nucleoside triphosphates complementary to the DNA nucleotides on the antisense strand, and shows that the sequence of nucleotides in the RNA is exactly like that of the sense strand, the non-coding, non-template strand of the DNA, except that uracil stands in for thymine. In this micrograph, we are looking at genes sections of DNA that are being transcribed. We cannot see the RNA polymerase molecules, but there are many of them moving along the gene in this direction, synthesizing RNA molecules that are dangling to the side, seen here. As the RNA polymerase moves along the DNA in this direction, the lengths of the RNA molecules increase. Numerous RNA molecules are being synthesized at the same time, and they are increasing in length as the RNA polymerase moves along. This makes the entire structure appear like a feather. Here is a diagram that helps explain the previous slide. RNA polymerase begins here, transcription begins here at the promoter, and moves along the DNA, synthesizing the RNA polymers that increase in length as the RNA polymerase moves further and further down the gene. With many RNA polymerases working simultaneously, there is the appearance of a feather. So here's an IB syllabus statement that demands more detail in your understanding. Describe the process of transcription, including the role of the promoter region, RNA polymerase, nucleoside triphosphates, and the terminator region. The template strand of the gene, the antisense strand, has a promoter region that allows the RNA polymerase to recognize the gene, to recognize the location where transcription should begin. Then RNA polymerase transcribes the DNA sequence just downstream of the promoter, with nucleoside triphosphates arriving to be added to the growing polymer, RNA polymer, in a 5-3 direction. The tail end of the gene will have a terminator sequence that brings transcription to an end. So remember, nucleoside triphosphates arrive to the active site of the RNA polymerase, and the two phosphates are cleaved off to form the nucleotide, which is here, that's added to the growing RNA polymer. The energy in the bonds between phosphates provides the energy required in the anabolic synthesis of the new RNA polymer. Here we can see the arrival of a nucleoside triphosphate that's complementary to the DNA template here. These two phosphates will be cleaved off to form the cytosine nucleotide. And the next nucleotide will be added to the 3 end of this nucleotide. Transcription happens in a 5-3 direction. Here's the next nucleoside triphosphate arriving. We can see the bond formation between nucleotides. The RNA polymer is synthesized in a 5-3 direction against a DNA template strand. And the bond between the nucleotides has formed with the cleaving off of two phosphates, providing the energy to form this bond in the formation of the RNA polymer. So here's a strong outline of transcription. The RNA polymerase binds to the regulatory region of the template strand known as the promoter. Hydrogen bonds are broken, DNA unwinds. RNA polymerase synthesizes mRNA. Nucleoside triphosphates are converted to nucleotides as they're added to the growing RNA polymer in a 5-3 direction. Base pairing rules apply. The terminator region stops transcription. Here's a relevant IB syllabus statement. State that the promoter serves to regulate transcription and provides an example of non-coding DNA with a function. The promoter region of a gene is sometimes called the Tata box because of the sequences of thymines and adenines that characterize it. The promoter region is in red here, signaling the RNA polymerase as the location to begin transcription. Transcription is initiated here in blue. In this image, you can see the RNA polymerase bound to the promoter region of the DNA, and I've identified the orientation of the anti-parallel DNA strands. And then I ask, if strand 2 were the template strand, describe the orientation of the newly synthesized RNA strand, and in which direction would the RNA polymerase move, left to right or right to left. The RNA would be 5-3 anti-parallel to the template strand. The RNA polymerase would move left to right as new nucleotides would be added to the 3 end of the growing RNA polymer. In this diagram, I've identified the promoter region in red and the transcriptional unit downstream of the promoter. Here we see the RNA polymerase having arrived and having unwound the DNA and the newly synthesized RNA growing as the RNA polymerase moves left to right along the gene. Note the anti-parallel nature of the template strand and the growing RNA polymer. So while we've been talking about the regulation of transcription by the presence of the promoter, let's look at other factors that regulate transcription. Outline the structure of nucleosomes, state that nucleosomes help supercoil the DNA, state that nucleosomes help to supercoil chromosomes and help to regulate transcription. The supercoiling of chromosomes serves to regulate transcription of DNA to mRNA because when the DNA is supercoiled, it cannot be transcribed. I've introduced nucleosomes in previous movies. The supercoiling of DNA during condensation regulates transcription. When the chromosome is tightly coiled or condensed, transcription cannot occur. This explains why there is very little protein synthesis during mitosis. The linker sections between nucleosomes, right in here, are more likely to be transcribed than the DNA wrapped around the histone proteins. Unique or single-copy genes are more likely to be located here and here and here rather than as a part of the DNA wrapped around histones. So are you making the connections as you move along? Hopefully, because here are two IB syllabus statements relevant to the connections that you should be making in the transcription story, but there's more to come. So I want you to see these two syllabus statements so that you can keep your eyes open. First, can you outline 4 regions of DNA that do not code for protein? Now you know of the promoter, and you know that regions of DNA wrapped directly around histone proteins would also not be transcribed. But what about regions near the centromere? And there's more to come, so pay close attention. The second statement outlined 4 mechanisms by which transcription is regulated. The supercoiling of the chromosome and the promoter regions influence the binding of RNA polymerase. Stay tuned for more. With this slide, I would like to introduce you to some very interesting ideas on the regulation of transcription. In this strand of DNA, there are 4 genes, a regulatory gene, and 3 others that are adjacent. The regulatory gene codes for a protein that regulates the expression of these 3 genes by binding to the DNA here in a region adjacent to the promoter called the operator. When this protein is bound to the operator, transcription stops. Thus, in addition to supercoiling having an influence on the regulation of transcription, certain proteins play a role in regulating transcription as they influence the expression of other genes. These 3 genes, called structural genes, are transcribed as a single mRNA unit. The mRNA would be processed so that 3 separate proteins would be the end result. All 3 proteins would play a related role in a metabolic pathway. So we do have genes that play a regulatory role and genes that result in proteins that determine the visible appearance, the phenotype of the organism. When we're thinking about regulatory genes, consider once again the concept of one gene, one polypeptide. Regulatory genes do not produce protein that directly influences the visible appearance of the organism. All of the genes that we can see here in this image being transcribed by RNA polymerase may be regulated by the presence of promoters, by the presence of regulatory proteins, and by methylation, a process we're about to see. So I've come back to this IB syllabus statement, outlined 4 mechanisms by which transcription is regulated. You know of promoters. You know of proteins that bind to specific base sequences upstream of the gene, the promoter or the operator. Nucleosome supercoiling limits transcription, and methylation of DNA. Methylation? What's that? Methylation is a slight change to the DNA described as adding methyl groups, CH3 groups, to the DNA. And when DNA is methylated, expression of genes is inhibited. Methylation is a slight change to the DNA described as adding a methyl group to a DNA base. We can see methylation here along this DNA strand, and when the DNA is methylated, the expression of genes is inhibited. When we add methylation to the DNA strand, genes get switched off. The fascinating aspect of methylation is that it changes during the lifetime of an individual depending on their life experiences, for example, experiencing trauma. Experiences in the lifetime of an individual alter the methylation of the DNA, and methylated DNA can be inherited. We'll be doing some reading about methylation in class. Epigenetics is the study of genetic variation caused by external environmental factors that switch genes on and off. The environmental factors are experienced during the lifetime of an individual. Now, patterns of methylation are established during embryo development and reach a maximum at birth. The amount of methylation decreases throughout life. If a pregnant mother experiences traumatic events or wonderful events during her pregnancy, this could influence the expression of genes in her child. Identical twins have nearly identical methylation at birth, but methylation differences accumulate over their lifetime. Environmental factors influence the pattern of methylation and thus influence gene expression. And DNA methylated over the course of one's lifetime can be passed to their offspring. So here are the IB syllabus statements relevant to methylated DNA. State that methylation of DNA bases serves to regulate transcription. Analyze changes in DNA methylation patterns. State that the environment of a cell has an impact on gene expression. As the last concept in the topic of transcription, heading towards translation, we need to look at the post-transcriptional modification of RNA, known as RNA processing. In this diagram, you can see the raw mRNA transcript here. An enzyme will remove sections of the RNA known as introns and splice together sections known as exons. This forms the mature mRNA. The enzyme that does this work is an RNA molecule known as a ribozyme. So here are the last IB syllabus statements of this movie that are relevant to the modification of mRNA following transcription. Explain the modification of mRNA following transcription. State that eukaryotic RNA needs the removal of introns to form mature mRNA. State the difference between introns and exons. The messenger RNA, once formed, is a raw transcript. It is not ready for translation. Sections of the raw transcript, known as introns, need to be removed, and sections of the raw transcript, known as exons, are spliced together to be expressed during translation. You can see in this diagram the introns are being removed and the exons are spliced together before the mRNA leaves the nucleus. In this diagram, we can see the chromosome with a small section of nucleotide bases known as a gene. There's a promoter region to which the RNA polymerase will bind. The gene could be coding for transfer RNA, for mRNA, or for ribosomal RNA. The raw mRNA transcript has its introns removed and its exons spliced together to form the mature mRNA transcript. I've come back to this syllabus statement because we can now add another region of the DNA that does not code for protein, and that would be the introns. We have the promoter region upstream of the gene to which the RNA polymerase will bind. We have introns. We have genes for tRNA molecules. We have telomeres at the ends of chromosomes which do not code for protein, and there are other regulators of gene expression. We've seen a bit of this with proteins that bind to the promoter or the operator of the gene to inhibit transcription. In this image, we can see the gene with its promoter having been transcribed into a raw mRNA transcript. The introns will be removed, the exons will be spliced together, a poly-A tail is added, a guanine cap is added, all of this to produce the mature mRNA transcript, which is then translated into a polypeptide in the cytoplasm. Once again, the raw mRNA transcript has its introns removed and its exons spliced together to form the mature mRNA. Further modification occurs with the addition of a poly-A tail and a 5' guanine cap. Through RNA processing and the splicing together of these 3 exons, this could result in the proper positioning of 3 functional domains in a polypeptide, such as the active site on one side of the molecule and the allosteric site on the other. You may have wondered, does RNA processing have any functional significance? And the answer is yes. A single raw transcript through alternative splicing can result in different polypeptides being formed. At this point you should say, what? Wow, that's amazing. Here's the IB syllabus statement. Describe how alternative splicing of mRNA increases the number of different proteins an organism can produce. So a single mRNA transcript with different exons can be alternatively spliced, notice the color coding here, into different mature mRNA molecules, each of which would result in a different protein. Here's an actual example of alternative splicing in humans. The same gene in the cells of 2 different tissues, thyroid and hypothalamus, the same mRNA transcript in 2 different tissues, results in different exons coming together to form calcitonin in the thyroid cells or CGRP in the hypothalamus cells. So as we wind things down here, it's important for you to remember that there are regions of the DNA that do not code for protein, the promoter region upstream of the gene, introns, genes that code for tRNA or rRNA, telomeres at the tips of chromosomes, centromeres, with which you should be familiar, and regions of DNA that are wrapped around histone protein. In this image, there are 4 regions of the DNA that would not code for protein: the centromere, the telomeres at the tips of the chromosome, the promoter upstream of the gene, and the introns, which are removed. It might have occurred to you that the regulation of gene expression can occur at numerous stages in the process of producing polypeptides. Regulation can occur at the transcription level, or at the RNA processing level, or at the translation level. The more steps there are, the more opportunities for regulation of gene expression. And that brings us to the end of IB Bio Transcription Translation Part 2. In Part 3, the focus will be on translation.